All right. I guess we'll begin. So, thanks everyone for tuning into first Funk lecture of the year. Woo! Before we begin, got a few announcements. So we currently have a series of workshops being offered. The next one is going to be in late February, and we'll announce three more in the upcoming weeks. And these can all be found on our website, biogeography.org, and you can easily register there. The registration fee is slightly cheaper for society members. And if you're not a member and you want to learn more about the benefits, you can find this all on our website. The next biannual conference will be in our house, Denmark, in January of 2026. And later this year, we should have more on conference registration and abstract submission. And at the conference, there are two main awards, the Alfred Wallace and the MacArthur and Wilson, that recognize its outstanding contributions to biogeography. And if you would like to nominate someone for either award or self-nominate for the MacArthur, the deadline to do this is this Saturday, February 1st. And to learn more about the nomination requirements, you can go to our website, and underneath our What We Offer tab is where you can find all this information. And then at the end of the talk, we'll have some time for Q&A, so you can type a question in, or if you want to speak directly, you can raise your hand and we can unmute you. And yeah, thank you for attending. This is being recorded and we'll post it on YouTube shortly after. And I'll pass it to Marta to introduce our speaker.
Thank you, Raymond. Yes, it is great to be back after the holiday break. We had a couple months off, but we're back to it and with yet another amazing Funk seminar planned. So it is my pleasure to introduce our speaker today, Dr. Jamie Wood. Dr. Wood is a senior lecturer in environmental genomics at the University of Adelaide and the Australian Centre for Ancient DNA in Australia. Dr. Wood received his bachelor's and PhD from the University of Otago in New Zealand. Until 2022, Jamie was a senior scientist at Manaki Wenua Landcare Research, which is one of the New Zealand's Crown Research Institutes. In 2022, he moved to Australia for his current position at the University of Adelaide. Dr. Wood's research applies evolutionary and environmental genomic approaches in combination with other techniques such as palynology, macrofossils, radiocarbon dating, stable isotope analysis, and so on, to study the composition and functioning of terrestrial ecosystems over the past 50,000 years. And today, I think we will hear more about the topic. So without further ado, Jamie, the floor is yours. That looks good.
Okay, excellent. Thanks very much for the invite to give today's Funk lecture. What I'm going to be talking about is the ghosts of species interactions past, so the biogeographic legacy that these past interactions between species have left. So you've already heard a little bit about myself, but this is where I've been based since doing my undergrad. Did my undergrad and PhD at University of Otago before moving to Landcare Research in Lincoln, where I was part of a group working in paleoecology, particularly palynology and ancient DNA. And as Marta mentioned, just 3 years ago, moved to the University of Adelaide, where I'm now part of the Australian Centre for Ancient DNA. So I really regard myself as a paleoecologist and not a biogeographer. So I feel a little bit out of my depth giving this sort of talk, but I think there's a huge overlap between these two disciplines. And my interests lie in how biodiversity and species distributions have changed in the past. And to do this, I use a range of different approaches, including ancient DNA, environmental DNA, bone isotopes, and palynology, and I'll talk about some of these today. So when I was asked to present this month's Funk lecture, I looked back at some of the previous lectures that had been given and was actually really impressed to see how much paleoecology has featured. Okay, so just going back over the past few years, there's been talks on things like the ecological legacies in Amazonia, right back to Mesozoic plants. There's been talk on paleoecology, landscape dynamics, changes in small mammal communities, and ancient DNA. Okay, so these topics in paleoecology have been really well covered. So, and what they've really done is demonstrate the importance of and value of these long-term ecological records in terms of understanding current day species distributions. And so I really thought about, what's something new that I could bring to this lecture, and how can the past help inform the present in regards to biogeography in a slightly different way. So much of what we do in paleoecology is using fossil evidence to construct species presence or absence at different time points at different places. So whether this is, for example, reconstructing plant communities using pollen analysis of sediment samples, or whether it's using bones from cave sediments to look at past mammal communities, or perhaps even dung fungi spores to track local abundance of herbivores. By doing this, what we're doing is reconstructing community composition. And if we do this for a number of samples of different ages, we can reconstruct how those communities change through time, such as shown here in this diagram. So on the left, we have kind of a classic pollen diagram figure where we're going up through time, and we can see the changing composition of plant communities. And on the right, using dung fungi spores to track how local abundance of large herbivores might have changed through time. So by using a range of different proxies, we can begin to reconstruct not only community composition, but entire ecosystems. So we can detail all the animals, the plants, the fungi, the microbes that coexisted at a particular place and time. But I guess what I'm really interested in, and what a large part of my research over the past decade has focused on, is reconstructing the interactions between those species. So who was eating who was pollinating who was dispersing who, and so on. And these details can really help us understand the functioning of a past ecosystem. And it can also enlighten us as to the potential impacts of extinctions of species within that ecosystem. So if we remove one of those species, then how might those connections lead to impacts on other species within the ecosystem? And I think this has significant relevance to biogeography. So when we think about biogeographic patterns and species distributions, we tend to think of environment and climate as being the key drivers, but actually species interactions are really important as well, whether it be competition, predation, or parasitism, or some other interaction. These can have a big impact on where we find species. And species interactions, I would say they're not easy, but they're relatively easy to study in today's world. Okay, so we can go out, we can observe species, we can see what they're eating. We might be able to do DNA analysis of scats and find out what they're eating. But how do we study interactions between species that may no longer be around, or which may no longer co-occur because of range contractions? But they might have relevance to understanding current day biogeographic patterns. So in today's lecture, what I want to do is draw upon some of the examples from our work in New Zealand to try and illustrate some of the ways in which studying the past, and in particular species interactions, can shed new light on our understanding and provide insights into past ecosystems, but also present day ecosystems and potentially ecosystems into the future. But before I begin, I just want to kind of set the context and give a little bit of background information about New Zealand for those of you who aren't familiar. So New Zealand is really just the emergent part of this recently discovered eighth continent called Zealandia. So in the diagram here, this outline we can see is basically the edge of this continent, Zealandia. So this is a significant piece of continental crust. Most of it is now beneath the ocean. And Zealandia's had a very dynamic geological history. So if we go right back to the Mesozoic, it was actually part of the continent Gondwana. Around about 80 to 70 million years ago, it separated from Gondwana. And as Zealandia moved away, it began to sink. Okay, so the crust got stretched, it got thin, and it started to submerge beneath the ocean. And this continued right through until about the Oligocene, where pretty much most of Zealandia was beneath the water. And this is a major event that's referred to as the Oligocene drowning period. Shortly after this, the plate boundary moved back into the area now occupied by New Zealand, and you can see that on this map as this kind of large feature running north-south through New Zealand. And this renewed tectonic activity actually began uplift. So it sort of brought more land above the ocean. And so this dynamic geological history has really had a major impact on shaping the evolution of New Zealand's flora and fauna over time. Overlain on top of this is this recent dynamic climate history. So during the Pleistocene and Holocene, we've had these periods of glacial interglacial cycles that have had a huge impact on the flora and fauna. So the vegetation of New Zealand has fluctuated between forest communities during interglacial periods and then non-forest habitats during glacial periods. In terms of macrofauna, perhaps the most interesting aspect is this apparent lack of terrestrial mammals and the corresponding dominance of birds in the macrofauna. So New Zealand, when it broke away from Gondwana, was at a time before placental and marsupial mammals had evolved. So there was no sort of early mammals on the New Zealand landmass as it split away. It was dominated by birds. And this bird-dominated fauna has a number of characteristics. So these include really ancient lineages of bird that were present probably at the time of separation from Gondwana and have kind of been relict in New Zealand for the last 80 million years. But it also includes recently dispersed species coming from Australia. The genetic analysis of a lot of these species has shown that some of them evolved gigantism really rapidly. So in this picture, what we've got is some of these large flightless ratites, the moa, and they're being hunted by this giant eagle. And this eagle, genetic analysis has shown, is most closely related to the little eagle in Australia. But when it flew to New Zealand, it became giant really rapidly, like in a matter of a few 100,000 years. So it happened very rapidly. And it was driven by isolation and occupying a new niche where there were a lot of these large birds around. Another characteristic of New Zealand's fauna, unfortunately, is high extinction rates following human arrival. So New Zealand was the last major landmass on Earth to be settled by people. And it was first settled as part of the East Polynesian diaspora. So around about between 1025 and 1120 AD, people moved into the Society Islands area. And then shortly after a small hiatus of 100, 200 years, there was this massive expansion of people out to all the islands in East Polynesia. So everywhere from Hawaii out to Rapa Nui and to New Zealand. And as with all the islands that were settled at this time, New Zealand, the landscape was transformed by people. So significant tracts of forest were burnt, especially east of the Southern Alps, this mountain range that runs north-south. So on the east of that, there's drier conditions, rain shadow, and those dry forests burnt very quickly. And together with hunting and the effects of the introduced Polynesian rat, the New Zealand mainland and its offshore islands lost around about a third of the bird species that were present. But more locally, this was more severe. So in the North Island of New Zealand, for example, up to half of the bird species were lost. And we really know a lot about the history of New Zealand's biota because we've got really good fossil records. So, and these records include things like wetlands. So New Zealand wetlands have gone through massive transformation as well. Around about 90% of them have been lost. But those that exist preserve these excellent pollen and charcoal records of past vegetation history and also fire history. New Zealand also has really extensive bone deposits and perhaps some of the best late Quaternary fossil deposits anywhere in the world. And as I mentioned, during the Oligocene, New Zealand was virtually completely submerged. And during this time, a lot of limestones were forming. So when New Zealand got uplifted, those limestones provided the perfect geology for forming caves. So right across New Zealand, there's extensive cave networks and sinkholes where large flightless animals have fallen in at different times in the past. And here we can see one of these cave deposits from the west coast of the South Island, just piled up with these bones of large extinct flightless moa. So these types of fossil deposits can inform us about what species were co-inhabiting the same environments. And also by analysing bone isotopes, we can get information about the trophic level of these species, and I'll talk a little bit more about that shortly. But also within the past couple of decades, these vast accumulations of coprolites have been discovered. So previous to about 20 years ago, only a handful of coprolites had been found in New Zealand, and it wasn't really possible to say with confidence what species they were from. Some of these large ones, we could pretty safely say were from moa, but there were 9 different species of moa living across New Zealand, all with different ecologies. The discovery of these large deposits of coprolites fortunately coincided with the early stages of ancient environmental DNA research. And this actually made it possible to use DNA sequencing to identify which species of bird each of these coprolites had came from. And so this really paved the way for a much more in-depth picture of species interactions, because these coprolites contain the remains of species that were eaten, but also the parasites of the animals. And this photo shows one of these coprolites from an upland moa, which is one of the 9 species of moa. And so for the rest of this talk, I'll discuss some case studies of where we've actually been able to explore species interactions and some of the insights that these have given us in terms of the evolution and the biogeographic history of New Zealand's flora and fauna. So I'm going to start off by talking about these really strange birds, the adsbills. So they're in the family Aptornithidae, and these are gruiforms. And they're a deeply endemic lineage in New Zealand. And so there are fossil remains. So here's a fossil vertebrae of an aptornithid from about 18 million years ago. And this is virtually identical to the adsbill vertebrae that were around just before humans arrived. So for at least 18 million years, these birds had been large and very similar morphologically, and it seems likely that this lineage probably was in New Zealand at the time that it separated from Gondwana. Genetic analysis of the Adsbills has suggested that their closest living relatives are probably the South American trumpeters. And these are frugivorous birds that still live in South America today. The Adsbills were flightless. They have these very reduced wings, and they're very large. Okay, so they stand up to about 31 inches in height, weighing up to about 40 pounds. So these are big birds. They're flightless. And so their extinction was probably through the process of hunting in much the same way that moas went extinct following human settlement. For a long time, there's been a big question about the ecology and in particular the diet of adsbills. As I mentioned, they're closely related to these frugivorous birds from South America. But Richard Owen, when he analysed the skeleton of these birds back in the 19th century, suggested that they could actually be omnivorous. So he saw features that indicated they might have been eating animal material as well. The first evidence for diet of adsbills actually came from a comparison of dietary stable isotopes, so delta 15N, delta 13C, from a number of different species. And so in this plot here, we can see that we've got delta 15N on the y-axis, delta 13C across the x-axis, and here we've got 4 species of New Zealand bird. Down the bottom, we have the stout-legged moa, a herbivorous species, and also a herbivorous duck. We've got the New Zealand owlet nightjar, so an insectivore, and then the North Island adsbill. And the key thing here is that the delta 15N is really an indicator of trophic level. So the more enriched a species is in 15N, the higher its trophic level. So this really sort of indicated that the adsbills could have been very high up the food chain. The issue here is that all of these samples are from different localities and ages across the North Island. And this can complicate isotopic analyses. So to give you an example of how this can complicate things, here's a plot showing delta 15N values from 2 species of herbivorous moa. So we know that these birds are strictly herbivores. And if we look at the crested moa, we can see that the 15N values change through time. So as we're moving between glacial and interglacial times, this can actually have an effect on the 15N values. And for heavy-footed moa, if we look at these very recent specimens, we can see that they exhibit a broad range of 15N values from about 9 right down to about sort of 2 or 3. And what this reflects is not necessarily differences in diet, it's differences in aridity or moisture availability. So when we use these isotopes to look at trophic levels, what we really need is a single well-constrained site where the bones have been deposited over a very short time period. And this allows us to control for these potential complicating factors to do with climate. And we're really fortunate to have such a site in the Canterbury region. So this is Finch's Folly. It's a tomo, which is a sinkhole in limestone. And this particular tomo trapped thousands of different birds. There's literally thousands of bones from this site. And it has 2 useful things from an isotopic perspective. So one, as I mentioned, it's a single site, so that limits spatial variability. But also the bones have been deposited over a very short time window, literally over just 1,000 years, and that limits the variability to do with climate. So the isotopes here should provide a really unbiased view of trophic levels. And if we look at where the birds plot, so again, down the bottom, we've got three different herbivorous species. We've got a finch's duck, geese, and a kakapo, which is a herbivorous parrot. Above those, we've got some omnivores and insectivores, tuatara, which is a reptile, the weka. And then again, we can see that the adspills are coming out right at the top. So this really suggests that these adspills are essentially top predators. So they're not only eating herbivores, they're actually eating other omnivores potentially as well. And this result was further corroborated by an analysis of isotopes from another site, so Harwood Flat near Dunedin. And here again, we've got a single site, and again, it seems to be very constrained in terms of the time over which these bones were deposited. So here, we've got two different moa species. We've got actually Polynesian dogs, and dogs, their diet can be very variable. So they can have almost a strictly herbivorous diet right up to a meat-dominated diet. But we can see the adspill coming out with these really elevated delta 15N values, again suggesting that it's acting as a predator. If we look at the distribution of adspill remains across the South Island, so these red dots are where adspill bones have been found, and we overlay a pre-human vegetation map, we can see that the distribution of these bones is really restricted to these dry podocarp forests that existed east of the Southern Alps. And are absent from these regions such as Central Otago, where we really only had grassland or scrub habitats. So these seem to be a bird that were living in forest. And I mentioned before about this giant eagle that New Zealand had. It had the largest wingspan of any eagle known, and it probably inhabited these non-forest or forest margin habitats. So it might have been the adspill that really filled this role of a forest floor predator in New Zealand. And why is that interesting? Well, if we look at what other bird species occurred in New Zealand forests, we can see that a lot of them had these unusual characteristics when we compare them to birds from other parts of the world. So we have birds that exhibit very high rates of nocturnal activity, birds that exhibit camouflage plumage, and also a lot of birds that exhibit burrowing behavior or that lay their eggs in small crevices. So why exhibit these behaviors if not to avoid predation? And New Zealand, when we think about New Zealand being a land of birds tend to be visual predators. So things like camouflage and nocturnalism work really well as predator avoidance strategies. Less so against introduced mammals, where scent is an important thing for prey detection. So perhaps these adspills were a key reason why these traits became so well represented in the New Zealand avifauna. And one of those birds that was driven to camouflage, nocturnalism, and utilizing burrows was this bird, the kakapo. But before I talk about the role the kakapo has to play here, case study 2 is really about a plant. And this is the wood rose, or Dactylanthus taylori. It's a very unusual plant. It's parasitic, so it lives on the roots of forest trees. And the only visible part of the plant are these flowers that emerge through the forest floor in autumn. Pollen records show that this parasitic plant was once widespread across the North Island. So the area we can see in green here is where we think Dactylanthus occurred at the time of human settlement. But now it's found only in these black areas. So it's been reduced to about 4% of its former range. So it's a highly threatened plant. It's eaten by introduced possums and rats. And it's also been harvested for decorative purposes by people, especially in the 19th century. So there are conservation plans around managing these last remaining populations of Dactylanthus, and a key conservation management strategy is translocation. So being able to take plants from these existing populations and move them to new sites to kind of expand the range of the plant. But these are really limited by one key factor, and that is the pollinator of these plants. So this is the lesser short-tailed bat. It's one of only two terrestrial mammal species found in New Zealand. The other one is another bat as well. And this species is the only known pollinator of Dactylanthus. The problem is the short-tailed bat is itself a very highly restricted species. So it occurs mainly as small populations that are sparsely distributed through the North Island. And this really limits the potential places that Dactylanthus could be reintroduced to, because there's no point putting a plant somewhere where there's nothing that's going to pollinate it. A few years back, though, we made a serendipitous discovery while looking for Kakapo coprolites in the Honeycomb Hill cave system. So the Honeycomb Hill cave system is a very large cave system. Again, it's in limestone, and it houses significant bone deposits that record the fauna of this area. But it also has Kakapo coprolites that span the last 2,000 to 3,000 years. And you can find these still sitting on rocks within the cave system. And they provide a record of what Kakapo were eating over that time period. So we collected a number of these coprolites, and here's one we analyzed. We made a pollen slide, and when we were looking at the pollen that was present in that, we found these really unusual pollen grains, very distinctive, and we quickly realized that they were from this Dactylanthus plant. And they had quite a high proportion of these pollen grains in the coprolite. So when we wrote this up in 2012, we said that this relatively high abundance, so we found about 8.9% of the total pollen in this coprolite was Dactylanthus. It's a zoophilous pollen type, so it's not blowing around in the air. The only way it could have got into that coprolite was through a direct feeding interaction between Kakapo and Dactylanthus. A larger study of more coprolites by Alex Boast recently supported this idea. So it found a high proportion of coprolites that were studied contained Dactylanthus pollen. And in some of those coprolites, up to almost a third of the pollen content was Dactylanthus. So this indicates that what we found was not just a one-off occurrence, but that Dactylanthus was actually being targeted by Kakapo. And it makes sense because Dactylanthus flowers in autumn, and this is a time of year when other food resources may be quite scarce and limited. And these flowers also produce large amounts of nectar that would be very appealing, not only to Kakapo, but potentially to a wide range of other bird species that also utilize nectar. And so this realization through analyzing a single Kakapo coprolite has raised the possibility that the plant might not actually be pollination limited in its range through the lack of bats, as was once thought, but that actually many native bird species might utilize the nectar as a food resource and could act as potential pollinators as well. And since the study was published, it's actually been confirmed through video monitoring that other birds will visit and feed on the nectar of these plants. And so it raises new possibilities in terms of potential conservation translocation sites. So case study 3 involves sequestrate fungi. So these are fungi that hold on to their spores. They're not like mushrooms where the spores can go out into the air and get blown around. These types of fungi, such as truffles and other truffle-like fungi, retain their spores within the fruiting bodies. So we're quite familiar with things like truffles being these dark and dull coloured fungi that we see on the right-hand side here. But New Zealand actually has a number of these truffle-like fungi that are brightly coloured and they emerge above the forest floor. And we can see an example on the left here. Around about 30 years ago, a New Zealand mycologist, Ross Beaver, said that these colourful and emergent truffle-like fungi seem to be overrepresented in New Zealand. And he came up with the hypothesis that this was due to birds. So these fungi were essentially mimicking fruit. On the right-hand photo here, we've got these classic truffles that occur beneath the ground. And these are dug up and consumed by mammals. And mammals have a really good sense of smell. They can locate these when they're buried beneath the soil, dig them up, consume the truffles, and then disperse the spores via their dung. But New Zealand doesn't have mammals, and birds are very visual in terms of finding food. And so Ross Beaver's idea was that these truffles had adapted for being dispersed by birds. And we can see in the photo on the left here, we've actually got two of these fungi on the right-hand side of the photograph. You can see the stalks. But the other objects in the photo are actually fruit of mero trees that are occurring in this forest. So you can see that there's a very strong resemblance between the fungi and the fruit. Now, this is a fascinating hypothesis, but actually no one had ever tested to see if this was the case, and in particular, whether these colourful truffle-like fungi were overrepresented in New Zealand compared to other parts of the world. So a few years ago, Amy Martin undertook a postdoctoral project to explore this issue and test this hypothesis for the first time. So Amy collated a huge number of distribution records for truffles and truffle-like fungi. And alongside that, she amassed a lot of information about the traits of those species, and in particular, the colours. And the analysis showed that indeed New Zealand did have a very high prevalence of these colourful truffle-like fungi, around about 80% compared to the next highest being South America at 65% and Australia at 61%. And in particular, reds and blues were highly represented in New Zealand compared to other land masses. So this study really demonstrated that the first part of this hypothesis was correct. So New Zealand does have an unusually high proportion and diversity of colourful truffle-like fungi species. But this is where the idea falls down through this kind of startling lack of any published observations of birds actually eating these truffle-like fungi in New Zealand. So as part of her research, Amy set up some video cameras, and in this photo we can see on the left-hand side this orange truffle-like fungi and a bird completely ignoring it. Okay, so it captured no interactions of birds eating these fungi. And it's kind of unusual because both birds and colourful truffle-like fungi are fairly obvious. And so it seems peculiar there would be so few instances of recorded consumption. But then we started thinking about New Zealand's high extinction rates, particularly of large ground-dwelling bird species, and we wondered if perhaps dispersal could have been largely facilitated by some of those species that are no longer around. In 2018, Alex Boast used some 18S DNA sequencing to identify plants and fungi in coprolites of 4 moa species. And a number of different fungi genera were identified. And interestingly, 2 of these sequences were attributed to Cortinarius. And this is a genus that includes many of these truffle-like fungi that we're talking about. But the thing is that Cortinarius fungi are also ectomycorrhizal, and so their DNA is likely to be abundant through the forest soils, and it could have got into the moa coprolites through moa ingesting soil when they were eating or grazing low plants on the ground. So what we really need is a combination of fungi DNA and spores to demonstrate consumption. And when we went and looked at pollen slides from these particular coprolites, we failed to find any spores that could be attributed to these fungi. Now, a couple of years back, when Alex undertook his PhD research, he again made quite a serendipitous discovery that provided this much needed link between species. Alex was looking for kakapo coprolites, and at two localities, Hodges Creek and Takahe Valley, he found coprolites that DNA sequencing actually showed weren't kakapo, but were from an upland moa, which is a small species of moa. And DNA analysis of these two coprolites actually revealed they contain DNA of a number of fungi that were these truffle-like fungi species. And what's more interesting is when Alex looked at these spores that were present in these coprolites, he found spores that could be directly attributed to these fungi species. And some of these spores were abundant and tended to be in big clumps, which is exactly what you see when you look at dung of modern mammals that consume these fungi. So it demonstrated a direct link between moa and these fungi. So now we can demonstrate a high representation of colourful emergent truffle-like fungi in New Zealand compared to other land masses. We can show a direct link between extinct moa eating these truffle-like fungi. Where does this leave us in terms of understanding current and future biogeographic patterns? Well, it turns out that many of these truffle-like fungi are mycorrhizal, so they form a really tight relationship with the roots of many native tree species, and these include forest dominants such as beeches, so Lophozonia and Nothofagus, as we can see in this photo. And the ability of forest margins to expand into non-forest habitats often relies on the dispersal of mycorrhizal spores into those habitats. And this role is largely performed by mycophagous animals in other parts of the world. So the worry here is that if the key dispersers of these fungi have now gone, New Zealand forests might have a reduced or slower ability to respond to environmental change in the future. So this could actually be a case of an extinction legacy, where the true impact of an extinction within the wider ecosystem has yet to be fully realized. So the examples I've talked about so far involve trophic interactions, so predation, pollination, and mycophagy. And lastly, I just want to briefly touch on a case of parasites. So coprolites are not only a source of information about what a species has eaten, but also the parasites that live with that species. And this can be in the form of eggs or larvae that can be viewed on microscopic preparations of the coprolite, or through DNA analysis of the coprolite. And in this study from a few years ago, we looked at the parasite egg content in 84 moa coprolites from several different species and locations. And a subset of these coprolites were also used for DNA analyses. And based on the results of this, we were able to create host-parasite interaction networks, which revealed some shared parasites between moa species, but also some that appeared to be host-specific, so we only found them in one particular moa species. And interestingly, a subsequent study of parasite DNA from both moa and kakapo coprolites failed to detect any overlap, suggesting that the majority of parasites that infected moa may have been specific to moa. And this kind of makes sense given their distinct phylogenetic distance to any other birds. And so I guess the interesting thing about studying species interactions, and in particular things like parasitism and mutualisms, is that there's a certain degree of reliance on each species. And in the case of parasites, they rely on having their host species. And this really underpins this process known as co-extinction. So co-extinction it's a secondary process, but despite this, it's thought to be the number one driver of biodiversity loss, with more species being lost as a result of co-extinction than any other direct cause. But co-extinction, despite being such a major contributor to biodiversity loss, is a really poorly understood process. So estimates of co-extinction rates tend to vary widely, and they're largely based on modeling approaches that come with a number of underlying assumptions. And I think the real difficulty with studying co-extinction is that it potentially occurs over very long timescales. So the decline of a host species may take decades or even longer, and parasite species may be lost along the way. So I would argue that in terms of long-term records of parasite change, such as what we can find in time series of coprolites, that these can provide some much needed empirical data on this poorly understood process, and one which may play such an important role in biodiversity loss. So just to finish up, a couple of take-home points. First off, fossil evidence for species interactions does exist in several forms. It's not very common, but it is out there. And in particular, late Pleistocene and Holocene animal coprolites can provide these exquisite records of species interactions, including diet and parasitism. Insights from studying past species interactions can provide a context for understanding modern biogeographic patterns, evolutionary histories, and importantly, potential future trajectories of ecosystems. Thank you.
Thank you very much, Jamie. That was a great talk. If you have questions, please put them in the Q&A box, and I will read them off to Dr. Wood. I don't see any questions in there now yet, but hopefully they will arrive in a second. But as we're waiting, I have a question because I was listening to your talk and I was really fascinated by the third case study on truffle-like fungi. So I know that you said that there's no evidence for the extant birds to be feeding on those fungi at the moment. Do you think any of the invasive mammals can be feeding on that? Any other group, any other taxonomic group that might be filling that niche that used to be filled by, what now is extinct birds?
Yeah, that's a really good question. And there's very limited data on that. So we undertook a study a few years back where we studied what types of fungi invasive mammals, and in particular deer and possums, were consuming. And we did this through DNA analysis of scats and camera trials. And what we found was that they were largely eating invasive fungi, but that these native truffle-like fungi did occur in the droppings as well. So they are eating them. We then did a inoculation trial. So we took seedlings of some of the native beech trees and some invasive trees as well. And we actually applied those droppings to the soil that those trees were in. And what we saw is that we got really good inoculation of those seedlings by invasive fungi. So the mammals were eating invasive fungi, and those were mycorrhizal on the invasive trees, and that seemed to work really well. But actually, none of the native tree seedlings that we inoculated with the dung formed mycorrhiza. So even though these mammals were eating the native fungi, I don't know what was happening, whether it was something to do with the digestive processes that were going on in the mammals. Those fungal spores did not seem to be viable and able to infect the seedlings once they came out. So it may not be a very simple process, and I think it needs a lot more research. We do know that the mammals are eating them, but whether or not they are providing effective dispersal is something we need to look at a bit more.
So that's interesting. So some of the fungi that are eaten by mammals come out as viable
Yeah,
spores, not the native ones.
they tend to be the introduced fungi. Yeah.
We have plenty of questions from the audience. The first one is from Suzanne Renner. Fabulous talk. I am puzzled by the fungus eating only having evolved in New Zealand. Why not in other island systems with forest floor birds?
Yeah, it's a really good question. I think we did find evidence of that. So it's not that these colorful emerging truffle-like fungi are only found in New Zealand. We did see them across a number of other parts of the world, and in particular, South America and Australia had, good numbers and high proportions of these colorful fungi. So they do evolve in different parts of the world. But perhaps, these places that had mammals, there was less evolutionary pressure, if you like, to do that. And so while some did go that way, there were still mammals around to find and consume those fungi that were under the ground. In New Zealand, there were no mammals to do that. And so essentially, if you were a truffle-like fungi and you were under the soil, the chances that something would find and eat you were very, very limited. So there was a greater pressure, I think, to evolve those adaptations.
Makes sense. Thank you. Another question is from David Hembry. Some studies have suggested that double mutualisms, in which the same species of animal serves both as a pollinator and seed disperser of a given plant species, are unusually common on oceanic islands. Has any of your work found examples of this in the New Zealand biota?
Good question. Yeah, I think we certainly have a really good handle on dispersal, seed dispersal, particularly of moa. So we've looked at a large number of moa droppings, and we understand what seeds pass through intact into the droppings. And we also have a lot of evidence of pollen in the droppings. But I think the really difficult thing, and it's something we struggled with a bit in the Kakapo study, is how do you prove pollination using these samples? We know that there's large amounts of pollen in there. It indicates that the animal was feeding on a flower, but we're trying to discriminate whether that's feeding on nectar or pollen or just eating the entire flower, which is not super useful for pollination, is kind of a really difficult thing. And so what we rely on is having these kind of coprolite analyses to generate hypotheses that we can then go and test in reality. So in the case of the Kakapo and Dactylanthus no longer occur together. And so that was really difficult to test. And it just so happened that there was a few Kakapo that were put somewhere where there was these plants around. So we're able to set up cameras and actually see what they did to these flowers. But yeah, I think certainly in New Zealand, it would have been the case. I think we had very generalist birds. They would have been acting as pollinators and dispersers. It's just very difficult to prove that using the samples we're analyzing.
Great, thank you. Another question from Camille Gallard, which might be related to the first question. Thanks for the stimulating talk. Could you elaborate on the explanation behind the uniquely higher prevalence of colorful truffles in New Zealand? Why did it happen there, and why didn't it happen elsewhere?
Yeah. Well, I think it's probably a unique combination of having the forests that have these mycorrhizal fungi and having this bird-dominated fauna that really sort of drove the prevalence there. And I think in other land masses, you might have one of those aspects, but not necessarily the two combined. So I think it's just a unique thing to do with sort of the biogeographical history of New Zealand that's led to that.
We have time for one more question, and there's one from an anonymous attendee. Wonderful talk. The other birds that were found to feed on the nectar of wood rose, have any of them been found to serve the same role as a pollinator like the short-tailed bat?
Well, so there's been very limited camera work done on dactylanthus plants, and that's probably one of the reasons why these bird interactions haven't really been seen. But certainly the video that was taken of a small passerine bird, it was really putting its face right into the flower. So it was after the nectar, and there's almost certainly no question that it would have got a lot of pollen on its face afterwards.
Well, thank you very much. I don't see any other questions in the Q&A. Let me check the chat. Yep, nothing in the chat. So thank you very much, Dr. Wood, for joining us. I know it's really early in Australia, so we appreciate it that much more that you made the time to join us for the seminar. And we will post the video of the seminar on our YouTube channel. So if any of the attendees want to go and check it out again, please do so. It should appear there within a week or so. And yeah, well, please help me thank Dr. Wood, and till next time. Bye, everyone. Bye, Jamie.